Hello everyone, welcome to this VINPAI webinar. We are very happy to have you with us today. My name is Vincent Talvas and I'm in charge of the Asian market at VINPAI. I'm here today with Hervé Demey, the scientific director of VINPAI, and Brieg Malegol, the person in charge of the Middle East market. Today's presentation will be focused on our clean label solution that we combine the algae and the fibers together. So the focus today will be divided in two parts. Firstly, Hervé will introduce us all the natural benefits of algae, and in second part, myself and Brieg will show you, will demonstrate you all the potential you can get with the clean label solutions we developed together, and we will make some demonstration and show you the applications we did. So I hand over the speech to Hervé.
Thank you. Well, welcome everybody. This morning I will show you a presentation about algae, and the title of the presentation is Algae, the Unexpected Obvious Evidence. So, through this presentation, I'll explain to you why and how the oceans and algae, in particular, are an essential resource to meet the food challenge of the 21st century. So, let's start with the first slide. The title of the slide is The Sea Source of Life. Well, maybe you know, but oceans have seen the birth of life on Earth and have contributed, thanks to algae, to its development on emerging continents. They cover more than 70% of the surface of our planet, and they contain 97% of the available water. They're also home to an animal and plant biomass which is estimated to 30 billion tons. And due to the very short life cycle of most of these species, the average annual production of this biomass is around 5,030 billion tons, which is to be compared with the 3.5 billion tons of food biomass which is produced annually by agriculture. So, no doubt that with millions of animal, plants and microbial species, the sea is undoubtedly a great source of blue technology. Next slide, please. So, we will focus this morning on algae as a resource for nutrition and health. Why? One of the facts is that algae are the fastest growing plants in nature. Please. Next one. In fact, algae have an ability for some of the species to triple their biomass every day. That means that 1 acre of cultivated algae can produce annually the same amount of protein as 21 acres of soybeans or 50 acres of corn. And to do that, algae don't need arable water, lands or fresh water, which is very important for tomorrow. By doing this, algae also produce a lot of oxygen. They are the main provider of oxygen on Earth, roughly 70%. And like most of the living organisms, they produce a lot of things, and mainly sugars like polysaccharides, proteins, lipids, minerals, and fibers. Next slide, please. So, when talking about algae, what are we talking about? We need some definitions. Algae, in fact, is a polyphyletic group of very different organisms, but which have in common some traits. The first one being the chlorophyll, with oxygenic photosynthesis. The life cycle of algae is generally realized in aquatic or humid environments, and mainly salted water. Algae don't have roots, nor flowers, nor seeds. And the size varies from 0.5 micrometer to 70 meter. This group of algae include both prokaryotic and eukaryotic organisms. The prokaryotic organisms are the blue-green algae, which are in fact the first photosynthetic organisms which appear on Earth roughly 3 billion years ago. And the eukaryotics means organisms with nucleus, divided in microalgae, which are very small, unicellular organisms, and the macroalgae, which are multicellular. So, when talking about algae, there is an English word which is very easy, which is the seaweed. What are seaweeds? Seaweeds are specific algae which are microscopic, multicellular, and marine. And in this group of seaweeds, we have three main families, which are the green, the red, and the brown algae. And the green are in fact the ancestors of the terrestrial plants. So we start with the oldest one, the green seaweeds, which are, as I told you before, the ancestors of the terrestrial plants. They are roughly 1.5 billion years old, and there are some common traits in between themselves. The first one is the color, the green color, which is due to the presence of chlorophyll A and B. And one other very specific point, which is the type of polysaccharides that we find in those seaweeds, which is in green seaweeds, the ulvans. The second family is the family of the red seaweeds. They are a sister group of the plants and the green seaweeds on the same branch of the evolution. They are roughly 1.2 billion years old. In fact, their color, the red color, is due to the phycoerythrin and phycocyanin, which are accessory pigments to the chlorophyll. And in the red seaweeds, we find specific types of sugars, specific polysaccharides, which are the carrageenan and agar. So the next one is the family of the brown seaweeds. It's a quite recent group of seaweeds, only 150 million years old, if we can say so. And their specific color is due to some accessory pigments to chlorophytes, which are the fucoxanthin pigments. And the same of the other seaweeds family, the polysaccharides which we find in brown seaweeds are also specifics, and they are composed of mainly alginates and fucoidans and laminarins. When talking about seaweeds, we could consider that those plants are quite primitive, but in fact seaweeds have a unique type of metabolisms, and which are able to produce very diverse and unique chemical compounds. And starting with only the photosynthesis from the CO2 and the nitrogen from the atmosphere, those organisms are able to produce quite simple molecules, like, can you press the button? Yes, like basic sugars and amino acids, but also some more sophisticated molecules like glycosides. Can you press the button? Yes, and specific protein and enzymes, but also very, very specific molecules, quite complicated structure that can be found only in seaweeds. So why seaweeds? What is the interest of looking in seaweeds? Like in many other organisms, we find common molecules like protein, sugars, lipids, and of course minerals. But what makes the specificity of seaweeds is mainly the sugars, the polysaccharides, which can represent up to 70% of the dry weight. And what is interesting is that those sugars, the polysaccharides that can be found in seaweeds, are very, very specific and unique and can't be found in other plants or organisms. And mainly for the brown seaweeds, alginate and fucoidans, for the red seaweeds, the carrageenan and agar, and for the green seaweeds, the ulvans. And they are a source of a lot of specific properties, and especially rheological properties, but also a lot of different biological activities. But as you if you can come back, please. What is also interesting is also that they contains a lot of different types of molecules. And when talking about the proteins, they are also a very good source of amino acids because of a specific balanced composition of the proteins in the seaweeds. Also specific lipids. Most of the lipids present in seaweeds are long-chain fatty acids, and which have a lot of biological activities. And also the presence of very diverse minerals and trace elements, which represent, in fact, the variety of elements that can be found in the environment, their environment, which is the sea. Or maybe we can skip it. Next one. Just to show you that the production of seaweeds today on Earth is increasing every year. 40 years ago it was only less than 1 million tons of production, and today we have come to roughly 30 million tons. And why? Next slide, please. Because seaweeds are used mainly in human food as a vegetable. The second use that you know is the use of those seaweeds due to their texturizing properties, the food additives that are alginate, agar and carrageenan. Some of the use are as a nutritional supplement for the presence of protein, minerals and vitamins. The second important use of seaweeds is the industry, and especially the textile industry, once again using the rheological properties of the seaweed, which are the thickening, gelling or stabilizing properties. And a small part of the use of the seaweeds today is the use in the fine chemistry, in cosmetics and pharmacy. To summarize it, we know today that the main use of seaweeds is in the food, roughly 65%. The second use is also in food, but also in industry as colloids, and a small part of it is used in fine chemistry. So this slide is to show you the versatility of the polysaccharides that can be found in seaweeds. And this is the example of what we can find in red seaweeds, red algae. And red seaweeds contains mostly carrageenan, and you can see that depending on the chemical composition and the structure of the carrageenan, we have different types of carrageenan, kappa, iota, lambda, and then depending on the composition of the carrageenan in the seaweeds, we have different properties which can be gellifying, thickening or stabilizing, depending on the proportion of each of those carrageenan. Most of the carrageenan are soluble in water, but the solubility depends mainly on the presence of sulfate on the carrageenan. And you can see that the kappa carrageenan, which has only one sulfate, is much less soluble than instead lambda carrageenan, which has 3 sulfation on each structure. The presence of specific binding on the sugar, the bridge, which is called the 3,6-anhydro-D bridge, can also offer the structure the possibility to modify its structure and to form gels depending on the presence or absence of cations or anions in the environment. So a lot of those properties depend on the proportion of each of the sugars, and that means that depending on this proportion you can get some specific properties like jellification or for instance the formation of gels or the control of the water availability in the environment, which gives what we call the syneresis control. And to do that, when you formulate a product, you need to know what you want to do. And you can see on this slide that depending on the type of red seaweed that you use, you have different types of composition in the presence of the carrageenan. And so you can use one specific type of red seaweed, or you can use a mix depending on what you want to get in terms of rheological properties. So seaweed's solution for a better life, of course, as you can understand, by using seaweeds we use organisms which have very specific properties, and depending on the composition of what you extract from the seaweeds, you can get some rheological properties, and we can say controlled by the use of those sulfated polysaccharides, which are the alginates, which are the agar, carrageenan or ulvans, to form specific hydrocolloid and to get some properties in gel suspension and or AW control. But we also know today that seaweeds get a lot of biological properties due to presence of specific polysaccharides, also because of the presence and variety of trace elements, specific essential amino acids, specific fatty acids, and specific fibers, which, as you can see on the slide, can provide different biological properties: biostimulation, immune defense, antioxidant, antibacterial, antiviral, anti-stress, and so on. So, by the use of seaweeds, we can serve the whole food chain. Thank you for your attention.
Thank you, Hervé, for this great presentation. So, of course, during the webinar, don't hesitate to post your question on the chat during this webinar, and we will try to answer you the best we can afterwards. So now we know more about the seaweeds. We will show you how we use all these natural benefits of them by combining them with the vegetable fibers to develop properties such as texturing, stabilization, emulsification, and preservation. So how does it work? In fact, the solutions we developed became true regulators of free water. The fibers, they fix the water with their pumping effect, and the algae, they make the link with the main food components of the food matrix, like the fat, the carbohydrates, and the water, and therefore they maximize the pumping effect of the fibers. Indeed, only algae have such chemical compatibility with the food component. Therefore, several years of research in VINPAI led to the creation of two new products, the VIN'FIBRA and the VIN'EMULSIO. Next slide, please. So why would you choose these products in your formulation? In fact, the first question we should raise and ask ourselves is, what do consumers want today? Consumers today, they want more and more naturality in their food composition. They also want to have a clean label without chemical additives. They want to get rid about all the E numbers. And they also want the industrial to comply with their specific diets they are looking for, such as vegan, gluten-free, allergen-free, GMO-free. And to tackle this challenge, the industrials, they need as well to keep their price competitive and to maintain their all the organoleptic qualities they had before. So to tackle this challenge, industrials need to find the right formulation and the right products. Therefore, we truly believe that VIN'FIBRA and VIN'EMULSIO are the good solutions, the 100% natural clean label solutions you're looking for to answer these new market trends. So VIN'FIBRA and VIN'EMULSIO are made of different species and fractions of seaweeds and vegetable fibers. We dedicate more VIN'FIBRA to get good texturing, stabilization, and preservation, whereas VIN'EMULSIO is more dedicated is used as a natural emulsifier, and a substitute of yolk. We also recommend to not try to dilute those ingredients first in the water like you did with other products because we prefer to apply them and to integrate them in the formulation when the food matrix is already formed to get all the benefits of these ingredients. So that is to say we prefer to integrate them in the middle or at the end of the process. So we will now show you some applications. Brieg, I hand over to you.
Yes, thank you, Vincent. Thank you very much. So as you can see on the slide, we can make many things with VIN'FIBRA and VIN'EMULSIO. These two ingredients are very versatile and multifunctional. In fact, they are very universal ingredients. So you can use them in bakery applications like buns, cake, bread, biscuits, also dairy application like ice cream, cheese, margarine, cream. Also, you can use them in dressing sauces like mayonnaise, ketchup, tomato sauce, but also ready-meal products like vegan burger or yet. So we took the application of buns and we had this challenge to remove completely chemical preservative. So what we did, we took VIN'FIBRA at 1% in the recipe, and with VIN'FIBRA we reached 5 days of shelf life to compare with 6 days with calcium propionate. So it means the natural solutions brings already 83% of the initial shelf life. So how does it work? In fact, VIN'FIBRA reduces the free water content inside the product, and therefore the development of molds is greatly slowed down. So the algae and the vegetable fiber forms a complex which acts like a sponge which regulates the free water. So the buns lose less humidity content comparing to the control, and after 5 days you can see on the slide there is a gap of 1.3% humidity between the two products, VIN'FIBRA and with chemical preservative. So also during the baking the water is more diffused inside the buns matrix, and therefore the buns grow better, and you can see it on pictures with VIN'FIBRA. We see that VIN'FIBRA is more developed than the control. So during the shelf life, VIN'FIBRA is able to release the water fixed inside the food matrix, and it allows to improve the softness and the texture. Thanks to VIN'FIBRA and VIN'EMULSIO, we can remove all emulsifiers or chemical texturizers and replace them by natural ingredients. So what we did in this example, we replaced hydrocolloids, gum, starches by 1.5% VIN'FIBRA, and we replaced all chemicals emulsifiers by 0.5% VIN'EMULSIO. So how does it work? We've chosen algae species very reactive to lipids, and in combination with vegetable fibers, it distributes more efficiently the fat inside the food matrix, and therefore you don't need the help of artificial emulsifiers anymore. So in application like ice cream or cake, you can reduce the fat content by 25% without altering the organoleptic properties. So also VIN'EMULSIO can replace yolk in applications like mayonnaise or cake. For the dairy application, There's the fact that VIN'FIBRA is very compatible with dairy protein. So you can use VIN'FIBRA in dairy application like cheese, ice cream, or dairy cream, like also chantilly. So here is a recipe of processed mozzarella with almost 21% rennet casein, and we put 0.3% VIN'FIBRA. So how does it work? In fact, normally dairy protein is really degraded by the water content inside the cheese during the shelf life. So when we reduce the water activity, the stretch is mechanically improved. So the bond water, the water fixed inside the food matrix, the bond water inside the cheese helps to improve melting and browning. So when, for instance, we bake a pizza, the water evaporates less and it allows the cheese to melt well and to spread over the pizza to avoid from burning. And also, it helps to get a nice burning spot on top of the pizza. So we are going to show you the result on a real pizza. It's really coming from the cheese. It's a bit hot, but you can see already that the burning is nice. We have a nice spot with nice color. The cheese is well melted.
You want to cut a bit of the... We'll remove it. Okay, watch out.
If we can try to cut the pizza.
Smells good.
Smells good.
It's a bit hot. You can check the stretch.
Nice.
Look, it's nice. Well melted.
I'm already angry.
And you can already...
It's warm.
Should be hot.
It's a little bit hot.
Yeah, too hot. We'll
So
wait a bit.
In the same time, you can see VIN'EMULSIO and VIN'FIBRA.
Okay, thank you very much. Thank you for your attention. So now if you have some question, we can answer them. And I don't know.
Yes.
No question at the moment.
No question for the moment. Maybe you want to add some particular points on potential question
Yeah, if
some customers have.
some questions you could get is about declaration of the product. Maybe you can answer, Vincent.
Yeah, but the declaration, you just have to mention that the product, you have to mention the name seaweeds and vegetable fiber. It's really simple. Yeah.
Also, sometimes some customers ask about the color and taste. So as you can see, VIN'EMULSIO and VIN'FIBRA, it's a bit brownish.
A bit dark, yeah.
A bit green from the algae. So in application like dairy application like cheese or cream, dairy cream, you will put 0.3 to 0.5% to avoid any color in the product.
Here in this recipe you saw that you only put 0.3%.
0.3%.
Yeah.
In bakery application you go up to 0.7 to 1.5% in cake, in bread, in buns.
For instance, in France, we really love to eat spread seafood, and in those kind of recipes, you can go up to 2-3% without any problem.
And also, if you have seafood application,
Yeah.
you can go up to 2
Yeah.
to 4%,
Yeah. Right.
depending on the product.
Okay. Do you have something to?
No.
Okay.
No.
Thank you.
Okay.
Everything is perfect. Thank you.
Thank you
Well,
very much.
See you next time.
Thank you very much.
See you, and don't hesitate to come back to us anytime. We will be very happy to answer your questions and help you achieve all your goals. Thank you.
Have a good day. Bye.
Bye.
Bye bye.